This is TWIS. This Week in Science, episode number 826, recorded on Wednesday, May 26th, 2021. It's the little things that make the difference in science. Hey everyone, I'm Dr. Kiki, and tonight on the show, we are going to fill your heads with doom, deer, and just the gist. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Though you may be watching this from the comfort of your temperature-controlled home on your cutting-edge high-tech device while consuming your chemically, derivedly delicious sustenance, you are still simply an organism in an ecosystem with a role to play. Though you wear mass-produced coverings, mask your scent with artificial aromas, augment your appearance with pigments, and fill your days with mathematically created code, you are simply a hairless ape in a colony of the same. No matter how much we separate ourselves from the natural world, we will forever be a part of it. And no matter how much we try to make sense of it, it is simply the system in which we will forever participate. So if you want to hear these hairless apes chat about things which often cannot be explained, since it is far bigger than our very own existence, we are here to provide you some food for your overgrown frontal lobe. Tonight on This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about science.
Oh, yeah.
Yeah, because that's what we do. We come here weekly to discuss science news stories that we found fanciful, fantastic, fascinating, that piqued our curiosity. And we hope that this show does the same for you. Tonight, I brought stories about, what did I bring tonight? I've got a little bit of stem cell relaxation. I've got a gist. I've got the gist about a gist. And we have an interview with Dr. Elisabeth about scientific integrity and microbiomes. What'd you get for the animal corner, Blair?
Oh, I have climate change, bird poop, primate accents, and wolves.
Great. Lots of animals.
Mm-hmm.
Yep. A little bit of climate in there.
Yeah.
Okay.
It's all connected.
And a little poop, because
always
a little poop in the
Yes,
animal
as we
corner.
learned in our trivia, we talk about poop a lot on this show. So
We do.
You know.
We'll just put one more in the tally.
Yeah.
Okay, for those of you who are not yet subscribed to this program, you can find us by looking for This Week in Science on just about any podcast platform that's out there. Google, Apple, Spotify, Pandora, Stitcher, Spreaker, Radio TuneIn, all of them. We're out there. We're also on YouTube, Facebook, and on Twitch. Our website is twis.org. Time for more science. More science? The science. Okay. I have some headline stories that I want to start the show with tonight. Just really quick run down, because I wanted to hit a lot of notes. And I was just like, why don't I just knock them all out with just quick headlines? A meta-analysis of 338 studies of vaccine safety published in the journal Vaccine found that the vaccines we use, the MMR vaccine, diphtheria, pertussis, flu, all sorts of, name it, they probably were looking at it, we use are generally safe for kids and adults. This meta-analysis determined that aside from what we already knew were previously existing adverse events, that there, are certain individuals who do have a higher risk for certain events to happen, no additional risks were confirmed. Vaccines are safe. Just so you know. Mm-hmm. And on that note, the CDC has told doctors to be on the lookout for myocarditis in young people following vaccination for COVID-19 as a result of reports to the agency's vaccine unit. However, the incidence of these reports is not more than what would normally be expected in the general population. So this is something that has been noticed by doctors. It's been reported in the adverse events report system, but it isn't something that appears to be actually connected. They haven't made any connection to the COVID-19 vaccine specifically yet. But if doctors know to be on the lookout, they might find something we didn't know before. As for the effectiveness of the COVID-19 vaccines, the CDC is also reporting just 0.01% rate of breakthrough infections, even with increasing spread of new variants. This means that the vaccines are very effective at preventing infections of all kinds. So let's keep up the pace and save lives. Yeah, we can do that. But even if you do survive COVID and all the other diseases that are out there, how long can you really be expected to live anyway? Blair, what would you guess your life expectancy?
200, 250.
Yeah, that's your guess. Little Miss wants to live forever. Well, a new estimate of the potential absolute limit for human beings was based on blood cell counts and organismal resilience. It finds that the longest you can expect on this good earth is about 120 to 150 years. That's the limit.
I'll take it.
Yeah, that's what they say. They think we're going to get. But
I'll take 150.
I imagine there will be some battles with the anti-aging folk. And finally, that brings me to what you really have to live for, right? If you want to live 120 and 150 years, you've got climate change on the horizon, all sorts of issues related to that. But maybe, just maybe legal attempts will help the political and scientific efforts to draw down our massive carbon emissions. Dutch court ruled this week that the Shell oil company has until the end of the decade to get in line with the Paris Accord and cut its carbon emissions by 45%. I'm interested in seeing where this is going to go. How about you, Blair?
Yeah, that's awesome. It's more of that, please. We can't be trusted anymore to do things because it's the right thing to do. There have to be smackdowns if we're going to prevent catastrophe.
Right. I mean, that there has to be more regulation. And if the political will isn't pushing hard enough, fast enough, maybe legal efforts will do that. And the Dutch courts actually back in 2019 had another ruling that was based on a new standard, which basically comes down to don't do anything that is going to leave the planet worse off for future generations. And it's just a fantastic way to think, right? Yeah. But anyway, you had a story about climate.
Yeah. So it turns out maybe none of that'll matter because we'll all be infertile.
No. What?
Yeah. So it's very important to make sure that the current generation of living things survive. But if they can't have babies, it's a huge problem anyway, right? So this is a study looking at the impacts of climate change on fertility versus the impacts of climate change on fatality, basically. Mm-hmm. So if temperatures aren't hot enough to kill you, will it make you infertile? And so this is specifically looking at historical research, but now also this is research from the UK, Sweden, and Australia at fruit flies. So as with many things, we start with fruit flies. They have short lifespans, short generations. You get to see a lot of things in a short amount of time. They looked at 43 species of these flies to test whether male fertility was a predictor of global fly distributions rather than the flies at which the temperatures at which the fly dies. So that's their survival limit. And it did, in fact, follow that. So where you see flies on the planet has more to do with where they are able to still have babies rather than where they're able to survive. And so from this data, they estimated both the temperature that is lethal to 80% of individuals and the temperature at which 80% of surviving males become infertile. So these two different buckets, right? And they found 11 of the 43 species experienced an 80% loss in fertility at cooler than lethal temperatures. And rather than it recovering over time, it actually, infertility got worse over time after exposure. So infertility was more pronounced. There was more impact of infertility based on high temperature exposure 7 days after the exposure. And using that delayed measure, it looks like 44% of species showed fertility loss at cooler than lethal temperatures. So all of that to say,
you won't die immediately, but it'll be this slow decline
of the population.
The population.
So I feel like there's two ways to look at this. One way is it's really bad for all living things, or that maybe this is in the end the Earth trying to prevent us from overpopulating. It could be an interesting overcorrection in an accidental overcorrection. You know, the Earth's
it would be definitely accidental.
Cognizant. But it is interesting to think about how just socially birth rates are lower than in previous generations. Right. But it would be interesting. Again, this was fruit flies. There have been previous studies looking at mammal species and infertility with raised temperatures, things like cows, pigs, fish, and birds. So there is a historical significance to this looking at fertility research, but this could be a thing.
It could be, and it could just be one additional factor in not just human populations. I mean, if human populations can maintain with air conditioning and moving around, but our food crops and the species that we rely on, though they can't, then if ecosystems stop failing as climate change increases, then that's an issue. So
100%. Yeah.
It's all a system. But yeah, this does harken back to the New York Times article from, I think, earlier this week. Everybody's getting upset because there was a New York Times article pretty much about the decline of populations, that because of declining fertility rates, populations are going to decline, and that's going to affect the economy. And people have put that together before, but I don't think anyone had written about it in such a prominent newspaper previously.
Yeah. And I think that's just the big kind of flashing red lights that I wanted to bring up about this is when we talk about climate change's impacts on population dynamics, fertility is an important piece.
Huge piece. Well, speaking of fertility, what about stem cells? Stem cells are those wonderfully reproducible cells that can become, especially if they are the kind of stem cells that become any kind of cell in the body, the pluripotent kind, then they have much that they can do for us. However, governments have taken oversight and passed laws about when we can do research, what money can go toward research funding. And over a governing body, the International Society for Stem Cell Research provides guidelines to the research community internationally on kind of what research you should be doing, that you could be doing, and really helps people follow along the moral, ethical lines, which are not necessarily the same for every country, for every society around the world. And so this week actually, they dropped their new guidelines, and this is after a lot of conversation with many scientists, ethicists, people around the world putting these guidelines together. They have a new 3-tiered system for research into areas of concern, and the lowest tier 1, is the lowest level of concern, and into that tier is where they've put organoids, brain organoids. We've talked about them on the show before. Yeah, little mini brains, right? These little balls of nervous tissue that allow researchers to understand how brains work. But there have been questions raised about the potential development of consciousness by these organoids. Well, the researchers have all agreed that organoids are still at an early enough state of research that there's no chance any organoid is going to reach consciousness within about the next 5 years. So they've put it at the lowest tier of concern. Next level is level 2, and that is where they have put the transfer of human-animal chimeric embryos to a non-human uterus. So it's kind of like a middle level of concern. Okay, so it's a human and a mouse being put into, or a human and a rat cells being put together in an embryo and having that embryo put into a rat uterus, for instance. There are also human-pig chimeras that are being developed and others. The highest level of concern is anything involving the use of stem cell-derived gametes for human reproduction, the transfer of chimeric or model human embryos to human or ape uteruses, and the editing of germline genomes. And those are prohibited at the highest level of concern. So for now, the stem cell community is policing themselves and saying, let's not do this. We're not going to make human ape babies. That's not something we're going to do. We're not making human animal chimeric organisms that we will then bring to term. That is not going to happen. Everyone's agreed that is not something that the scientific community, stem cell scientific community, wants to do. Now, the final thing that they did, though, is they did drop their 14-day limit for human stem cell development. 14 days is just about when gastrulation starts getting started, and it's a really interesting phase of cellular and organismal development. Dropping the 14-day limit doesn't mean much for some countries. Like the UK actually has a law that says scientists cannot do research with human embryonic stem cells past day 14. That's it. The United States does not. So the fact that we don't have that kind of a law, now that this is kind of opened up, it does beg the question of what kind of research will this allow now? Or will the United States politicians, will they create a law because they don't agree with the allowance of further research? But it's open questions as to where this stuff will go. Cells in a dish is not the same as cells in a uterus, for sure. So it's an interesting state we are in, and it's wonderful that people are talking about the ethical aspects of where they want the research to go and how they want to manage it.
I want more evidence and logic-based rules for these things. So this is exactly the direction we need to move in. And I'm really happy that they dropped the 14-day limit.
It'll allow better understanding of early development, and it could really, really assist scientists. Yeah. So let's go from stem cells to your poop story.
Oh yeah, poop. And how we're all killing birds. Oh no. No, that's not actually true. So this is a study from Manchester Metropolitan University and University of Edinburgh looking at garden feeders in Scotland. So this is the big asterisk on this paper, is that it is specifically about birds and bird feeders in Scotland.
Got it.
So before we extrapolate this to the whole world, just keep that in mind. However,
this is not the whole world.
No, but it is a system that you can study for future research, right? So anyway, they wanted to see how garden feeders were impacting local populations of birds. And if you just want to see how a migratory population or a population that does not normally hang out where people are can run amok when you feed them, look at the Canada goose situation in North America. It's just a mess. They are not supposed to stay here year round. They're pushing other native birds out. There's all sorts of stuff, right? So they collected data on bird feeding in Scotland, just asking people, but also how else poop. So they collected fecal samples. They're always near bird feeders, and then they also looked outside of these bird feeders in local gardens and personal gardens. And they specifically were looking for peanuts. Why? Because peanuts are often found in bird feeder feed that's sold in Scotland, but is not a local plant. So,
right. That makes sense.
So that's like, okay, this bird was definitely feeding at this bird feeder. Also, just as some background, 150,000 tons of bird feed purchased in the UK, and that the amount of feed that they purchase is actually more than triple what all the birds would need to sustain themselves. So it's a little excessive. And they did a whole transect of Scotland. They were able to check all these woodland areas and specific gardens and backyards. And they found that 49% of samples had peanuts in them, but moths, which is the natural prey of these birds, were in about 34% of samples. So this is specifically looking for blue tit poop. And they found that it does look like the blue tits are getting the majority of their food from bird feeders. It also shows that as far as a mile away from a bird feeder, they found peanuts in poop. So they're coming from all over to those bird feeders and then dispersing back out. And this is a problem because there are increased populations of blue tits, and they are aggressive to other birds in the area, like willow tits. And so they've actually been known to evict willow tits from their nesting holes. So this is a case where this species is actually causing further problems for other species because it's too easy to live. So this is part of the conversation that we've had on the show previously when people have asked about feeding wildlife. This is why feeding wildlife can be really tough. And I'm not going to say all bird feeders are bad, because also sometimes like having a place for a bird to drink, if they're a migratory bird, can be really helpful if they're in an urban landscape that has chopped up what used to be wild, right? So I think that this is an opportunity for actually for biologists to set some parameters for what kind of feed, when to put out feed, and how much to put out. Maybe there are certain areas that bird feeding is not suggested because you're right next to a forest and those birds are taken care of. But this is just kind of an interesting first look at bird feeders and how we might actually be hurting more than we're helping, which can be kind of hard to hear sometimes.
Yeah, especially if it's helping the invasive species or the more aggressive species that push others out. Yeah, absolutely. But that's really interesting, as I have bird feeders at my house because I love the birds. I like to feed the birds, but sometimes I do wonder. Yeah, it's a great question.
It's a good question. And maybe in your area, you have some endangered species that benefit from bird feeders because you don't live in Scotland. So
that's right. I did wake up to a northern flicker at my window the other day, which was a nice sight. But anyway, moving from poop to the microbiome, I have one story before we get into our interview. Scientists from over 60 countries have published a sweeping analysis, and I'm using it as a pun because they swept surfaces for microbes, and they looked for genes for antimicrobial resistance, and they wanted to see kind of what species are out there in urban areas. So this is 60 countries and multiple continents. We're talking about the urban areas they looked in, sampling city surfaces and the air for microbiome evidence. They found 31 species present in 97% of the samples. So out of some 4,700 species that they did identify, there were 31 that are everywhere in every urban area. Every urban area also had a fingerprint, so to say. Researchers at Weill Cornell University who were in charge of the study, they say that based on a swab of your shoe, they could probably pinpoint where you live within about 90% accuracy, which isn't, 100%. It's not as good as, say, the Pfizer or Moderna vaccines, right? But 90% efficacy to identify where you live based on the bacteria in your urban area. They're hoping that they'll be able to figure out more about where antimicrobial resistance exists, where it doesn't exist. Maybe also not have to find genes from various organisms in the rainforest. How about a bus stop? You never know what lives there. Maybe your bus stop is like the rainforest.
Wow.
Who knows? Who knows? This is This Week in Science. Thank you for listening. And if you are enjoying the show, please tell a friend. I would like to take this moment to introduce our guest to the show, Dr. Elisabeth Bick. Some have called Dr. Bick a vigilante, but she's a PhD-trained microbiologist and has studied a variety of microbes at the Dutch National Institutes for Health, St. Antonius Hospital in Nieuwegein, I cannot pronounce that correctly, and Stanford University. In 2019, she left her role as Director of Science at Estart Medical in order to independently pursue scientific integrity. Welcome to the show, Dr. Bick.
Thank you so much for having me on. And I really enjoyed the talks about microbiome and poop, two of my favorite subjects. So
You're in the right place.
I fit right in. Yes. Yeah.
So I'd love to find out about your background. It's just fascinating the trajectory that your career has taken. Can you kind of take us on a little tour, and how did you get interested in microbes in the first place?
Well, I studied biology, and I basically had a fantastic microbiology teacher, and I really enjoyed the labs, going into the labs and smelling all these different bacteria that have strange smells. And most of my peer students did not like that. And I just love that smell of the agar being prepared, and just bacteria have different smells. And I realized that. And I also found it really fascinating that these microscopically small creatures could, grow in such amounts that we could actually start to see them on a colony, on a plate. And yeah, it was just fascinating. And it was right around the time when molecular biology started. So we could also learn about their genes, we could sequence them. And I was just hooked right away. And I studied microbiology. I did my PhD in microbiology and stayed in that field for a very long time.
For microbes, do you prefer bacteria, the fungi, or viruses?
I'm a bacteria girl. That's I found like viruses are a bit scary because they're not really alive, but they're also not dead, and they can make us sick. And it's just very scary when you think of it, that something that is not really alive can have such a profound impact on humanity as we are currently experiencing. But I don't know. I guess I grew up professionally with bacteria, and that has been my topic of study for a very long time.
And then you moved into scientific integrity.
Right.
What is scientific integrity, and how did you come to find it?
Well, scientific integrity is basically that as a scientist, you should remain honest and true. You should report what you're finding and not leave out certain amounts of data. You should not steal somebody else's ideas. You should not make up data. And so if you don't follow these rules, then you're maybe doing science misconduct. You're basically cheating, and you're lying about your results. And so I think that's a bad thing. I think scientists should be honest. And luckily, most scientists are honest. But because a lot of people in science are pushed to publish as many papers as we can, some people will try to break the rules and get more papers published by cheating, by making up stories. And so that is the part I'm interested in. And I think science misconduct can be tremendously bad for science, because other people might not be able to replicate those results. If people lie and cheat and publish that in a paper, then it's really hard for other people to replicate those results. And so it leads to a lot of waste of time and money. And it could even lead to people dying. For example, if you lie about the effect that a vaccine can have, as we can see now, then maybe people don't want to be vaccinated. And a lot of the mistrust about science and vaccines is based on papers that turn out to be based on misconduct. And so I'm very passionate about misconduct, and I feel there should be more people trying to correct science.
And when did you first kind of get started? I was doing background reading, and somebody plagiarized some of your work. Is that what started your adventure?
Yes, I was reading somewhere about plagiarism, and I just thought, just for fun, let's try to put one sentence that I had written and put it into Google Scholar between quotes. So I did that, and I had not expected to find anything else than my own paper. But I found something else as well. I'm like, what? Somebody stole my sentence. I wrote that sentence. I thought really hard about that sentence. I put a lot of effort in it. And then somebody just stole it and passed it off as their own. So I looked up that paper that had used my sentence and found out that they had stolen a whole paragraph of text that I had written. But not only that, they had also stolen paragraphs from other scientists. And so they made this new paper based on a patchwork of all different types of sentences stolen from left and right, and sort of glue that together into a new paper. And I was angry and I was fascinated, and I found more and more of these papers, and I wrote to the editors, and some of these papers got retracted. And so I felt, oh, I'm doing something good for science finally, because I've worked in a lab for a long time, and I just felt this was fascinating and very effective. And so that was about that was in 2013, and so I was doing that this sort of as a hobby. And then one day I found out by accident while I was looking through a PhD thesis that had plagiarized text. I also found an image that had a little smudge, something I recognized. And then in another figure in the same chapter, they had used that same Western blot, that was what it was, that same thing with a little smudge, but upside down, and it represented a different experiment. And I got even more mad. This person had plagiarized text and was also using Western blots, reusing them to represent different experiments. And this paper it was a PhD thesis chapter, but it had also been published as a paper. And so I wrote to another editor-in-chief of that journal, and that paper got retracted. And this was sort of successful, I guess. And then I decided to scan more and more papers to see if I could find more duplicates, because I realized I have this strange talent to recognize duplicated images.
And it's an amazing talent. I have been looking through some of the images, and I am so hard pressed to be able to recognize the patterns that you find within the images, and where, coming from an academic background and many people in writing in general, you learn to look for plagiarism online and you learn to look for the copied text. But to be able to identify the copied, not even whole images, but little parts of images, what are some of the lengths that people go to that you have seen in images?
Oh, there's all kinds of duplications that I found, and I do want to point out, I can only really find the tip of the iceberg. Only where people really leave traces of photoshopping, I would be able to find it. If a person is a really good Photoshop, and I should not really say this because people might now know my tricks, but a really good Photoshopper, I would not be able to detect that. So what I detect are duplicates, so duplicate parts of images. And so I sort of scan an image, and I'll let my eyes go over it, and then maybe I'll pick up on a duplicate. And I can remember maybe up to 20 or so images. So within a paper, I can look at the different panels, and maybe I can recognize and compare a couple of other papers. But I don't have the ability to remember all images ever published. I don't have any photographic memory or so. So it is limited what I can do. And I'm sure software in the end, which is being developed now, is going to do better. But as of now, I can do a decent job in finding duplicates between, within a paper, for example.
Do you think that your training in microbiology gives you some of that pattern recognition? Do you think did you spend a lot of time at the microscope identifying things when you were training, or do you have anything that you think you can put your finger on?
No, I did not really look a lot through a microscope, which is maybe surprising for a microbiologist, but I'm the microbiologist who studied bacteria by looking at their DNA, so sequencing and things like that. And so I'm actually very bad in using a microscope. I think I learned it as a college student, but I would not be able to get a decent microscopy images right now. So no, that's not something I would be able to do now. But I've always seen patterns. So when I go to a bathroom in a restaurant or so, I will look at the tiles. And I'm like, oh, that tile is the same as that tile, but it's upside down, or that tile is now rotated 90 degrees. And I do the same. I do the same with laminate flooring, where you have these fake photos of planks, and you sort of see the wooden pattern, and then you can just recognize that one plank is the same as the other plank. And I've always assumed that everybody would see these patterns, but I guess I'm sort of the odd person who will look at these things and is fascinated by them. And I'm now applying that for science. So it's a little bit more useful than studying bathroom tiles, for sure.
Although I'm sure you would be amazing at puzzles.
I don't think I'm pretty good at them. More, better than others. But I think maybe I have just the patience to look at hundreds or thousands of papers and look for these things. And I sort of see it as a puzzle. But then when people want me to play real puzzles, I don't know. I just don't really find that useful. I find it more useful to look at puzzles in scientific papers and do something. Wait a minute, I've got talent.
I can look through these images and I can actually do something here. Yeah,
right.
And you've started the hashtag Image Forensics on Twitter, which has gotten a really amazing following. Have you seen the community of people who are looking at images in papers grow in the years since you've gotten started in this?
Yeah, so I'm part of a small group of people who focus on published papers. So we get lots of tips, and we have our own leads that we follow. But I hear more and more from people who have found these things independently of that, who are just reading a paper for their work and finding a duplicate, or finding duplications while they're doing a peer review, for example. So I do think that the amount of people who recognizes these things is definitely growing, and I hope I've contributed a little bit by playing this game on Twitter and just opening people's eyes. Like if you know what to look for, you'll find these things in the papers that you read.
I know by following your feed and following your hashtag, I've tried my hand at things, but I haven't found anything in any papers yet. So, but if I do find something in a paper, your advice is to do what?
To remain calm and to not yell misconduct. So this is what I think a lot of people will immediately say, oh, this is misconduct. While in reality, a lot of these duplications might be honest errors. Not all of them, maybe only half of them. But it's hard to know from just looking at an image what really happened. So in a lot of cases, you can say that it's a duplicate. So what I would recommend is to write to the editor-in-chief of the journal in which you have found this paper, in this duplicate, and tell them about what you found. But remain very objective. So just say, figure 2B looks remarkably similar to figure 5C. Or there's an overlap with a little illustration. Unfortunately, I've done this a lot. I've reported around 4,000 papers by now. And there's, I know it's a lot. I've scanned like probably 100,000 papers, but I really only look at the images. So I don't read the papers. So I don't really read them. I should probably not be saying that, but I only look at the images. And so I have reported a particular set of papers, around 800, a couple of years back, 5-6 years back. And so I know what has happened to all these papers that I've reported to the editors-in-chief. And basically, with the majority of these papers, nothing happened. So I found these duplicates, I reported them, and then the editor did not take any action. So only in 40% of the cases did they either retract or correct the paper. So in the majority, there's just nothing that has happened to the paper. And I found that a disservice to the scientific readers of these papers. So these papers usually cost a lot of money. And if you're part of a university, your university pays a lot of money for these papers and these journals.
Yeah.
And the journals should care about the quality. So I feel that if a reader raises a concern about a paper, the journal should take action. If you buy a car and the wheel drops off a year later, you would probably go back to the dealer and ask, what's up with that, and can I get a new wheel? But it seems that a lot of journals don't really care about the quality of a paper after it has been published. So I'm taking things a lot online. So I either take them to Twitter, or I will, that's usually a sort of a last resort. But I post most of the papers on a website called pubpeer.com. And I will alert the reader that there's a potential problem with a paper. And then if people have a PubPeer extension, so they have plugins that will work with your browser, so you can do then a literature search. And if you have that plugin, it will tell you which papers have a comment. And you can click on that and see if there's a problem with the paper.
Interesting. See, I would think that a lot of these journals would respond to your message with a job offer, because I feel like it would be very helpful to have someone with your skill set in charge of publishing papers.
That would be wonderful, but no, I actually would like to remain independent, because I don't want to be working for a boss ever again. This is really good. It's just fantastic to not have somebody telling me what to do. But unfortunately, and it's changing. But let's say 5-6 years ago, when I started this work, most journals did not even respond. Or they would say, well, thank you, Dr. Beck, we'll look into this. And then I would never hear anything again. And I sent them reminders once a year, like what's up, any updates, and they would just not respond to those. So I'm taking things online, and I have even criticized big name journals for not responding to these things. And I think that is something that not a lot of scientists would dare to call out Science or Nature for not responding.
It seems like somebody needs to take the journals to task. The system, the entire scientific endeavor, the ecosystem, there's a lot of toxic practices that go on. A study just out on the big replication studies has found that even though these replication studies are going on to find out the power of the original studies that were done in psychology, it doesn't matter. People are still citing the original studies. There are no commentary. There's nothing going on to the original studies, giving them any kind of addendum or comment, or, just, hey, just a note, this other study was done that showed it wasn't valid, or giving it any sort of warning sign, I guess. But the publishers, they want to publish, and they're going to get money from libraries, and they're going to get money from people who are subscribing to their services. And then you have the publish, and perish within science. Do you see this kind of an effort as just one little step towards changing that ecosystem?
Yes, I have noticed some positive developments, but I also realize that scientific publishing, and I guess academia as a whole, is just an extremely slow moving ship. It's not going to quickly, swiftly change directions. It's going to take years and years. I feel there's a lot of things wrong with scientific publishing. Like, should peer review be done for free? And peer reviewers are not really educated to find fraud. They're educated, or they're supposed to find flaws in papers, but not necessarily fraud. And so that's not really their job to do. And editors will probably tell you that if they hire people like me who screen papers for image mistakes, and some journals actually do this, and that's a good development, but journals will tell you then we'll have to raise the price even more. And most academics will tell you, but we write the papers for free, we peer review them for free. Where does all that money go to? And because papers are not being printed anymore, so it is tough to see the amount of money we pay to academic publishing, and at the same time realize that there seems to be little quality control, not enough checks to check for these papers and check for these irregularities before publication.
Where do you think the drive to create paper mills comes from? Do you have any thoughts on why there are groups of people just publishing these fraudulent papers that are going out?
Yeah. So paper mills is a very, at least the ones I'm familiar with, are very specifically tied to medical doctors who work at hospitals in China. And that's not because people in China are more or less fraudulent than anywhere else, but it's because of a very particular requirement that these medical doctors, when they're done with their education and they want to work at a hospital, they have a particular requirement that they have to publish one scientific paper. But they're not given any time off to do research, and they might not work in a hospital that has time or a lab to do research in. So these are mainly people who work in clinical hospitals, not associated with an academic hospital or academic institution. So they're not their focus is to cure patients, not to do research. And so they're in a very hard situation where they have to publish a paper, but they don't have time or opportunity to do so. And so those people feel that the only way out of this, this situation they're stuck in is to buy a paper. And so there are companies called paper mills who will offer papers for a big amount of money. And then authors will buy their authorship into one of these papers. And these papers are completely fake, at least that most of the ones we're finding. The text is written through by ghostwriters on a template. The images are often stolen from other papers or are artificially generated, are not even real. And these papers are very hard to recognize as fake because each of these individual papers looks fine. I mean, maybe, not really high quality, but, decent. But if you compare a lot of these papers together, you will see that they have a very similar title structure, a very similar images. Sometimes they reuse images. And so it's only when you look at the bigger picture that you see all these that all these papers come from the same studio. And so we're together with a lot of other people, who most of them are anonymous. We're working on to find these paper mill papers and alert the editors that they should pay more attention. That there's just massive production of papers by almost like organized crime type of organizations that will pump out these papers.
That's fascinating that this is even happening right now. There were a couple of papers that came out. One paper, this is not so much paper mill as people publishing a paper with a particular slant. And I'm wondering whether it's to get attention or whether it's actually to give some kind of scientific credence to the beginnings of a conspiracy theory or some kind of idea that's meant to drive people's perceptions. And there was one study on that linked skin and 5G and COVID this last year that you had uncovered and blogged about. Can you talk a bit about what you found with that?
Right. Yeah. So that was, like you said, a paper that claimed that 5G, Gary, would produce coronavirus from our skin. And if your jaw is already on the floor, that should be. So this paper got published in a PubMed index journal. So it sort of looked legit at first glance, sort of passed all the quality controls. But if you read the paper, it looked impressive. It had, because it had a lot of formulas. Now, so I'm the type of biologist, if I see a formula, I'm like, I have no idea what it means, but I'm sure it's impressive. And I think that's what the peer reviewers thought. So it was full of formulas. And it's sort of, those formulas were supposed to show that 5G caused coronavirus to burst out of your skin. And somebody rightfully asked the question, well, if the 5G changes something in the cells of our DNA, the DNA of our cells, why doesn't it produce a $100 bill? Why does it specifically produce a coronavirus? And that, of course, that wasn't answered in the paper. And the whole paper was full of strange formulas and strange graphs but there was no data. And I always say, if you have a fantastical claim, you have to have fantastic data. And there was no data in this paper. And so I wrote a blog post about it, and the journal retracted it sort of silently. I think it's not a very good journal in which it was published, obviously. But somehow it passed all the quality control filters.
Looking into the authors, it seems the authors have published other very odd scientific claims in other papers as well. So there's a history of kind of pushing a fringe science perspective.
Right.
Anyway, I just wonder is something like that. The paper got retracted, but is something like that, is that just that researchers urge for attention, or is that something else? Do you think that there is the possibility with paper mills, with journals that maybe aren't great quality but get published in PubMed, that there might be the possibility, if we're not looking closely enough, of it being used to, like I said, give credibility to not credible ideas?
That's a tough question. And I think in this particular case, I would still blame it on some persons with strange ideas who found each other and just pushed out papers because they love doing that. I hope there's no agenda behind it, but I'm sure those papers will be misused by people who have either a very, like a strong anti-5G agenda. I'm sure they'll find this paper and they'll use it for that purpose. So I hope that it was not an organized action, but there's a lot of people with strange ideas, as we have all realized in the past year, and they will try to. They're convinced that scientists are not giving them a voice, and they'll find some topic, some platform to publish their ideas in. So whether or not that's completely organized or just the result of all the misinformation that we hear around us, and all the mistrust that people seem to have in science, that I don't know. But there's definitely a lot of strange papers that somehow have passed peer review. And I feel that we should be more on the lookout for those things, and we should be ensuring a better quality of publications.
So my question for the general public then would be, if I'm going to pretend I'm somebody who's not accustomed to reading scientific papers at all, how do I both maintain faith in the scientific process while also maintain a healthy skepticism of the information that I receive off the internet about the latest publication?
Well, usually I tend to look at the quality of the journal in which things were published, but even that is not a guarantee. We've seen some big retractions in big name papers in the past year. And so it's really hard to give 100% foolproof, this is what you should do to trust the paper. But in general, if things can be replicated, that's always a good sign. If, for example, the hydroxychloroquine paper that I wrote a big critique on, that paper has not really been able to be replicated by other studies. And so I think that's a sign that paper maybe is not a good paper. And but it's hard to give that. In general, I still believe that most science is to be trusted, but there will always be these odd outliers that fall through the cracks and that get out there and that just have not been peer reviewed very well. And that is why I'm a firm believer of post-publication peer review. I feel once a paper is published, you should allow other scientists to be critical of it and to write critiques about it. And so that's why I make a lot of use of PubPeer. So probably my guess would be go to PubPeer and see if people have written critiques about the paper and see if you can trust it. And that might be the only way to really advise it. But it's hard even for scientists to know what is true. Some papers just look real and turn out to be completely fake. But that is only a small fraction of papers.
There are so many papers being published right now, and that is really, it's the information overload. When the scientific community these are the people who have to read the papers, and if you are an expert in a particular niche area, read the papers in that area and tell other people what you think about them. Are they good? Are they bad? And you have to be willing to speak about them or criticize them if they are not good, so that that information can percolate out to other communities who maybe are not the experts, right?
Right.
Is there anything else that you want to say about scientific integrity and the work that you're doing currently that you think people should understand?
Yeah, well, it's not always easy to do what I do. I do receive a lot of criticism. So right now there is a threatening lawsuit, like a lawsuit that I might get because I criticize the work of a particular scientist. And so it is not always easy work. It could be even dangerous work. And I feel that I am a scientist who criticizes papers, but I don't try to attack the persons behind, the authors. I don't try to make it personal. And it's tough that I receive these ad hominem attacks, on the way I look or the way I talk or the way, my whole history. While in reality, we should be talking about the papers that I'm criticizing. And I feel that these lawsuits are sort of trying to silence me, and that's not how science should work. And I do hope, and I know that there's a lot of support for what I do, but it is a tough situation to be in. So it's I hope that will change in the future. That we'll be able to freely speak and criticize and build upon each other's papers without having to be afraid that we'll be sued for that.
What happened to the free scientific enterprise and
Right.
free inquiry?
I know that is what I was promised when I started this career, but it did not quite happen. Yeah.
Yeah. Well, thank you so much for doing what you do. And for our audience and people who are out there, you do have a Patreon account.
Yes, I am on Patreon. Elisabeth Big is my handle, I guess. Thank you.
Wonderful. You're welcome. Yeah, I think this is the work that you do is very valuable. And, until the AI come in and, the robots take your job,
right.
which potentially maybe that'll be a good thing if, if large databases of images can be assessed, more rapidly. But until then, your work is very important. And thank you so much for joining us on the show tonight. It's just wonderful to speak with you.
Thank you for having me on. It was my pleasure. Thank you.
You're welcome. Oh, and before you go, where can people find you?
So I'm on Twitter. So my handle is Microbiome Digest. And it's not microbiome with an E at the end because Twitter did not allow all the letters that I wanted in there. So I had to drop a letter and it became the E. So it's Microbiome Digest. But you can also search for my name. There's only, as far as I know, one Elisabeth Big on Twitter with an S, not with a Z. And that's me. So I hang out a lot. Way too much time on Twitter. So you can find me there.
Same.
Yeah, we all.
Once again, thank you so much. Have a wonderful night. I hope you get some rest from your work, and I will see you on Twitter again soon.
Yes, we'll see you there.
Thank you. Everyone, this is This Week in Science, and we are going to take a very quick break. When we come back, it'll be time for Blair's Animal Corner. Thank you for listening to TWIS. If you like this show, please consider heading to our website, twis.org, and clicking on our Patreon link. For Patreon supporters who support us at $10 or more per month, we will thank you by name at the end of the show. We really can't do this without you. Thank you for your support. And now it is time for the show section that has lots of animals in it. It's time for Blair's Animal Corner with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh, good. You said it.
Breathe. It's okay. Let's do that. We can do it.
All right.
Okay.
So if you're a monkey, if you're a tamarin, very adorable, tiny little monkey that lives in rainforests,
So
and you're in a new territory, you don't belong there. You haven't been there before. In fact, other tamarins live there. How do you get through safely? Well, if you are a pied tamarin, which is a critically endangered species, has one of the smallest ranges of any primate in the world, you might sometimes have to go through red-handed tamarin territory. They are found throughout the northeastern Amazon region. And when the other way around, when the red-handed tamarins enter the territory of the pied tamarins, other way. So these pied tamarins that have their teeny tiny territories, they don't want to give it up, right? When these red-handed tamarins that are all over the Amazon.
Wait. What tamarins? The first ones?
Pied.
Oh, pied tamarins. Tamarins with their teeny tiny territories. There was a lot of alliteration there. Okay,
yes,
thank you.
Teeny tiny tamarin territory. When the red-handed tamarins move into the pied tamarin territory, the red-handed tamarins adopt the calls used by pied tamarins. So this is the first asymmetric call convergence shown in primates. So that means this is the first time ever that has been recorded that one species chooses to adopt another species' call pattern to communicate. They're not merging. They're not cross-communicating both ways. Just one species going, don't mind me, it's just another pied tamarin walking through. And so there are a few reasons that researchers think this is the case. First and foremost, red-handed tamarins have greater vocal flexibility. So the likelihood that they could even do this is high. They also use calls more often than pied tamarins, so they just talk more. So that also would make you think that their brain would have more kind of elasticity around calls and stuff like that. So scientists believe that they alter their calls to avoid territorial disputes over resources. Unclear as to whether they're trying to get through unnoticed as a pied tamarin, or if they're trying to speak to pied tamarins in their own lingo. Could be either. But ultimately what we know is happening here is they all of a sudden are sounding like a pied tamarin when they enter pied tamarin territory.
That is so tricky.
Yeah.
That's sneaky.
Yeah.
I am respecting the tamarins a little bit more. I mean, the pied tamarin does look a little bit diabolical. So maybe you do want to fit in.
Yeah. I used to work with these tamarins, and they are wild creatures. And yes, they are critically endangered. There's so few of them left. But yeah, tamarins in general, I would encourage anyone who has 5 minutes and isn't actively driving right now to Google pictures of tamarins, because there really isn't a not interesting looking one. Every single type of tamarin is cute or weird or interesting looking in some way. So just as an aside, check that out. That's my little piece of advice to all of you on your internet wasting that you'll do, because everyone's going to waste some time on the internet today, guaranteed, right? Just look at tamarins. Anyway, point being, this is a really unusual, interesting way for species to communicate with each other. And yes, they are both primates. Yes, they are both monkeys. Yes, they are both tamarins. And yes, they are very closely related. But this is still very cool.
It's very cool. I mean, not just paying attention to a conspecifics call, but being able to mimic that call, being able to blend in, being able to, okay, it's the ventriloquist act, or the American going to England and pretending to be Canadian, eh?
Yes. Yeah, I remember for a while they were telling you, if you travel overseas, put a Canadian flag on your backpack. Anyway, yeah, it is like that. But it's also kind of more like walking through the zoo and making a call like a chimp as you pass the chimp exhibit, because it's a different species.
Yep.
So it kind of is like learning a language before you go into a country, but it's kind of more like mimicking the sounds of a language you don't understand completely and passing, which I think is really crazy.
So the question now is, do these animals understand the sounds that they're making? Right?
Do they understand each other? Do they understand the sounds they're making to the pied tamarins? Do the pied tamarins understand the sounds?
Yes.
That the red-handed tamarins are making?
I think that is a very important question.
Yeah.
Yeah. Is it just mimicry, or is it actually picking up the other language?
Right. And then I can't help but wonder, with our fairly poor human ears, when we go into a habitat and we think we're hearing certain things, are we always hearing those animals? Or sometimes are we hearing other animals mimicking those animals?
That's a great question. I don't know.
I watched
Now we
crows
gotta
fighting a red-tailed hawk the other day, and it didn't occur to me until right this second that crows are capable of a lot of sounds. Do you think a crow has ever mimicked a red-tailed hawk?
I have a starling that lives in my house that mimics so many. It mimics the crows. It mimics the song sparrow. It mimics the bird of prey that lives nearby. All of them. This starling mimics all of them. And so I have caught myself going, oh, I wonder if there's an osprey in the neighborhood. No, it's a starling.
Gosh darn starlings. So then you have to separate out mimicry versus communication. Is this mimicry that we're hearing in these tamarins, or is it communication?
Yeah. Yep.
Then when you look further out in the animal, which anyway. Lots
Which
to
one
unpack
is
here.
it?
Mm-hmm. Yeah.
Which one is it?
Which one is it? And moving from monkeys to wolves. Wolves in deer territory. Which one is it? Prey or savior? So this is a study looking at both Michigan and Wisconsin and the impact of gray wolves on deer populations. So specifically looking at Michigan's Upper Peninsula, wolves moved in the 1990s and 2000s. And what happened? Deer collisions went way down. So deer were getting hit by cars less. Coincidence? Good question.
So there are fewer deer.
Right? So another team of scientists gathered data about road collisions and wolf movements in Wisconsin. And they looked there and found that, in fact, what was happening was wolves were creating a, quote, landscape of fear. So once wolves colonize a county, deer vehicle collisions go down about 24%, which is not nothing. That is a good chunk. And it is partially the thinning of the population, which, as we know, is not always a bad thing. Sometimes there's too many deer and it makes deer sick. It reduces their resources. It's actually a problem for deer if there's too many of them. Mm-hmm. So that is one piece. But the other piece is behavioral changes. Wolves, it turns out, use linear features of a landscape as travel corridors. Things like, roads, also pipelines, stream beds, but they will use these linear pathways to move through a habitat. And so deer learn very quickly to avoid those linear features when wolves move in, which means they stay away from the road.
Wow.
So the presence of wolves obviously is looked down upon by ranchers because wolves eat livestock. And so there's all sorts of data being collected and being looked at the financial implication of livestock losses to wolves, because then we're stuck doing a cost-benefit analysis as a government of which is worse, the loss of the habitat or the loss of the livestock. Don't get me started on how we shouldn't be living off so much livestock anyway, but ultimately that is a conversation that the government has to have, because they have to decide, is it beneficial to keep the wolves? And this is why wolves were eradicated from most of the United States pretty early in westward expansion of Europeans, is because the wolves were getting at the livestock. But a 2008 study from the U.S. Department of Transportation estimated that crashes from hitting deer cost more than $8 billion annually. So if you can reduce that, then it actually might be more financially beneficial to let the wolves stay, even if they're taking some cows and sheep here and there. But the other kind of unknown ecologic or economic impact, which is what I always gets me stuck in. I get stuck in the fact that we're really not good at quantifying the cost of things, really, in an environment. But wolves are also reshaping ecosystems, as Justin loves to bring up. I know he'd talk about it if he were here, the whole Yellowstone situation, right? So there's a huge benefit to ecosystems. Very hard to measure that economically, But if you think about collisions, you have this $8 billion, but that's not even counting medical bills, fatalities, and other things that happen as a result of these collisions. This is really just looking at like if a vehicle's destroyed, if a road is messed up, if a roadway gets blocked for a period of time. It's looking at a lot of infrastructure-y type things. It's not really looking at the human element when it's looking at that cost. So this is potentially something that could be really helpful to know for wolf management and wolf advocacy in the United States. And just as a quick little
wolves
parallel,
are good for cars.
Yeah, turns out. In 2016, actually, there was a study done that found that cougars decreased the number of deer vehicle crashes in part of the eastern U.S. by about 22%. So a very similar number. So this is tracking with that same number, that same rough number. And so if that's the case, having natural predators around just by the nature of them being there can help alleviate this really this pretty big problem. And, you know, I grew up in the city. I didn't almost ever have to worry about hitting a deer until I started going to school in a more rural area for college. But there are lots of places in this country where people are scared to drive late at night or early in the morning. And it's kind of nerve-wracking because if you hit a deer, it could be the end of your car or the end of your life, potentially. It's really dangerous. So
Yep.
it's very good to know that there are natural solutions for this.
There are natural solutions to the problem of hitting deer with your car. Who knew?
Yeah.
And it doesn't involve going out and hunting deer necessarily.
Mm-hmm.
No.
Yeah. And
the
so
wolves do that.
Large carnivores have an ecological and economic value. That's really what this is about, is again, the whole ecosystem. You think about the whole system thinking, the package, right?
We get so tied up in our little details that we lose sight of the big picture and the whole system. And the system is essential, the ecosystem.
Mm-hmm. Systems thinking is pretty important.
Yeah, very important. I hope we get to go back to the Center for the Institute for Complexity, in Santa Fe. They do a lot of systems thinking there.
Oh, I can't wait to go back to Santa Fe.
Yeah. Yes. After COVID, we'll get to go back. Ooh, Sunday.
AC after COVID.
Yeah. BCAC. This is the new dividing line for modern society. This is This Week in Science, and I've got some stories.
Yay!
Woo, I've got a story about stimulating sight. Really interesting study. So we've talked previously about retinitis pigmentosa and how it leads to deterioration and of vision and eventual blindness. And there really are not many treatments for it. And researchers have been trying to come up with various ways to treat the disease, whether it's been gene modification or whether it's drugs or whether it's, hey, you're going to get a cybernetic eye. There's all sorts of things. And researchers just published from the University of Pittsburgh and the University of Basel in Nature Medicine on the first evidence of giving a person who has retinitis pigmentosa partial vision, partial recovery of vision with optogenetic treatment. And now optogenetic treatment is when we put light-sensitive molecules into cells. And I've talked about it on the show many times with respect to mice and controlling cells in mouse brains, getting mice to do different things. And usually it's a rhodopsin, it's a light-sensitive protein, a light-sensitive molecule that responds to blue light. But in this particular case, this 58-year-old man was injected with a virus vector, an adeno-associated viral vector that then put this channel rhodopsin crimson R into his eye. And it took several months of this vector to actually incorporate this new molecule into the light sensing cells in the retina of this man's eye. This molecule originally came from an algae, Chlamydomonas noctigama. And so now from algae to vision, this man went from not being able to detect any light at all to being able to at least locate on a table objects. He can't see them distinctly necessarily, but as opposed to being 100% in the dark, he's now able to make out shapes on a surface and be able to identify them. And the really interesting trick to this, though, is that even though this channel rhodopsin, crimson R, it doesn't respond to blue light, it responds to red light. And it won't just respond to sunlight, even though there's a bit of red in sunlight. There's not a lot. They have to put special goggles on the person. So the people who get this treatment have to wear goggles, and the goggles then take the visual signal and send red light and wash the retina in red light so that the photosensitive cells are then activated and allow the person to see. So it's a combination of molecular technology in addition to actual physical technology, goggles plus special chemical in your eyes, and suddenly you can see. Suddenly you can see. Would you, if you could get a chemical that would get rid of your color blindness, if you had it injected into your eyes, do you think that you would do that, or would you stick with what you've got?
100% yes, until you said the word injected into your eyes. I think that was tough because I won't even get LASIK because it scares me to have. I have a thing about my eyes. It creeps me out. But if I could get color vision back, it would make my life so much easier. I gotta tell you. Yeah, I don't think a day goes by that somebody doesn't use a color identifier for something to me, and I often have to be like, oh, I'm sorry. I don't know.
I don't see that color. Yeah. Well, this research is very cool. It's just a few people so far that they have started this trial with this optogenetic technique. And they've treated a few more people, found that it worked okay. They weren't able to do the face-to-face testing because of COVID. But they did determine that it was safe, that the dosage they were giving was safe. And they are now doing additional tests with increased dosage to see if they can get a better response with more cellular recruitment, with more of this light-sensitive protein getting into the retinal cells, and with better light detection and better vision. So maybe one day there will be people, and maybe they won't need goggles one day. Who knows? Maybe it'll be a different world.
Yeah.
And then the final.
So cool.
I know. It's amazing. This kind of science where it's like combining, it's very futuristic. I'm like, this is sci-fi.
I was sitting there thinking about having no vision at all, and then suddenly having the difference between light and dark and some just kind of crude shapes would be a complete game changer. It would completely change your life.
Totally. Yep. You'd be able to actually get around your house more easily. Like just basic tasks would be so much easier.
Easier.
Yeah. So more tests to come, and they are calling this new technique, this new kind of information. And the way that it worked, it took a long time for it to start working, and the subject had to start really learning how to see again. And so what they're calling it, the clinicians are now calling it with a whole new name, because no one's ever done this before. And so it's a new scientific field that they're calling visual rehabilitation.
Awesome.
Yeah. Brand new.
Awesome.
Field.
This is what the 2000s was supposed to bring. This is exactly this.
Just a little bit
limbs,
late, but we're here.
The artificial. It's not even that late. We're still in the first half. It's the artificial limbs that can interface with your brain, and this is exactly. I forget the flying cars. I don't need that. This is what we need.
I don't need those. Just give me all the adaptations.
Yes.
I need that extra thumb from last week's show.
Yeah,
and
totally. Yeah.
And my last story for the night has to do with memories.
Mm.
Oh, the details fade over time, don't they?
They sure do.
They just fade. And we know, we've heard from researchers through the years that if you access your memories over and over and over again, that it's that accessing of memories that maybe overwrites them a little bit and kind of muddies them up. But a new study that was just published in Nature Communications kind of explains the reasoning behind why they become less vibrant. And the gist is that we remember the gist. We don't remember the details that we don't need to remember. And so they had a number of individuals in this study look at these visual images, and they had to, they were prompted to recall certain aspects of the image that were either very detail oriented or more meaning related. So they had word image pairs, and they were supposed to say, for example, if the image was colored or grayscale, which is more perceptual, or whether it showed an animate or an inanimate object, which is more semantic or meaningful. And then they tested them right after they were trained, and then tested them again 2 days later. And they found that the participants, surprise, surprise, were faster at recalling the semantic details. The perceptual elements, just those were not, they're just perceptual, and they were not important. And the researcher says it's kind of like, imagine that you're reminiscing about a dinner with a friend before COVID. And you realize you can't remember what the table decor was, but you do know what you ordered while you had dinner with your friend. Or you remember a conversation with a bartender, but you don't remember the color of the shirt the bartender was wearing. Or you're like, it was a green shirt or a red shirt. I'm not quite sure. And they say this pattern towards recollection of meaningful semantic elements we demonstrate in this study indicates that memories are biased toward more meaningful content in the first place. And we've shown in previous studies this bias is clearly reflected in brain signals too. Our memories change with time and use, and that is a good and adaptive thing. We want our memories to retain the information that is most likely to be useful in the future when we encounter similar situations. So it's whatever means the most in the moment. What are you going to remember about tonight's show, Blair?
Probably something about aspects? Injecting into your eyeballs. Yeah, and also that all those pictures that our guest said looked the same looked different to me. I didn't have the eye for it.
You're not going to remember the color of different people's shirts, or
no. That's actually a thing. My brain doesn't store color because it can't trust it. I have a whole theory about this. So yeah, I have no memory for color. So that's actually kind of related to this, right? So
it is, absolutely.
So I do think my brain knows it's not relevant because it's probably not right. So if I assume something is a certain color, unless it's confirmed that it's a certain color, then so For example, I have really good memory for what color articles of clothing are when I am told. So when I go shopping or when I buy something and I come home, I ask somebody, what color is this? I have a really good memory that that sweater is blue because I was told it was blue. But yeah, in terms of just kind of like an environment that I'm in, I have no memory for colors at all.
I'd love to know what kind of details people out there have memories for. Do you have photographic memories, or are you more of a gist person like me? I'm more of a gist person. I'm a gist person. Anyway, science, it gives you the gist. And I think we're done with the show. Did we make it to the end, Blair?
Oh, we did. Look at that.
We did it. Oh my gosh. Who
Crazy.
knew?
Wow.
Who knew? Well, I do want to thank Dr. Elisabeth Bick for joining us on the show tonight. It was wonderful to have you on the show. And as a reminder to our audience out there, you can find her on Twitter. You can find her blog. We will have links at our website, TWIS.org as well. And oh my goodness, I guess it's time for me to say thank you to people. Do like saying thank you. Thank you, Blair, for a great show.
Oh, thank you, Kiki, for hosting.
And my shout outs to Fada for help with social media and with show notes, with Gord for manning our chat room, which may be changing soon. We'll have a conversation after the show. And Identity4 for recording the show. Rachel for your amazing assistance. And I would love to thank our Patreon sponsors for being our Patreon sponsors, for all of the support that they give us. Thank you to John Ratnaswamy, Kira, Carl Kornfeld, Melanie Stegman, DeKramsta, Karen Tazi, Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shu Brew, Darwin Handon, Donald Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack Bryan Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Mark Mazzaros, Artyom, Greg Briggs, John Atwood. This profile name is hilarious in the context of some other podcast. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you all for your support on Patreon. And yes, and if you are interested in supporting us on Patreon, you can head over to TWIS.org and click on the Patreon link. And on next week's show, we have an interview scheduled with Dr. Kate Clancy and Dr. Katie Lee. We will be talking about women's reproductive issues, their study into the COVID-19 vaccine and how it's influencing menstrual issues, and women in science. Should be a lot of fun. You ready, Blair, for next week?
I'm ready. I'm gonna get my pink hat warmed up and ready to go.
All right.
Yeah, I'm ready. We'll be back on Wednesday at 8 p.m. Pacific time, broadcasting live from YouTube and Facebook and from twist.org/live.
Twist.org/live. You know what I just did? I wasn't reading aloud.
That's fine. I'm still going to go. Want to listen to us as a podcast? Just search for This Week in Science, wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well. For more information on anything you've heard here today, show notes and links to stories will be available at our website. That's at www.twist.org. And you can also sign up for our newsletter.
You can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion.com. at gmail.com or Blair at blairbaz@twis.org. Just put TWIS in the subject line so your email does not get spam filtered into oblivion.
You can also ping us on the Twitter where we are at TWIS Science, at Dr. Kiki, at Jackson. Whoa, at Dr. Kiki. I'm going too fast. At Jackson Fly and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address or suggestion for an interview, please let us know.
And we'll be back here next week, and we hope that you will join us again for more great science news.
And if you've learned anything at all from the show tonight, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.